guys, welcome back to my channel Biomics. So in this video we are going to discuss about the processing of the pre-tRNA. So the tRNA which was during the process of translation, it is not in the mature form. First it needs to be synthesized in the form of pre-tRNA, then by some of the modification it is converted into mature tRNA. So let's see how this modification is taking place. So during the transcription there are some extra sequences which are present in this tRNA. Like in the 5' end there are some extra sequences, as well as in the 3' end there are some extra sequences. But this CCA sequence which are present, it is not by the transcription of the genomic sequence. It is being added during the process of modification of this tRNA. Okay, so this extra sequences which are present in the 5' end as well as the 3' end, that need to be removed by some of the nucleases. Here the nucleases are RNase P, which removes the extra part of the 5' end. Here the 5' end, here the extra sequences are present which is being removed by the RNase P. And the 3' end is also having the extra sequences which are removed by other nucleases, which is known as RNase F or RNase E. These are the endonucleases. These are the endonucleases. So after this endonuclease work, it leaves some of the sequences which are present near the 3' end. These are removed by another nucleases which is known as RNase D. This RNase D, what does it? It trims this nucleotides. Okay. Then by the use of ribonucleotide transferase, there is some modification. That means there is addition of CCA group in the 3' end where the amino acid comes and bind. So now let us see the splicing of pre-tRNA. So there are exons as well as introns which are present in the RNA. So the exons are the coding part. So the introns needs to be removed for the formation of a mature tRNA. So for these steps they require certain nucleases, phosphodiesterase, kinase as well as ligase. So first, for the removal of this introns, this needs to be cleaved. This introns needs to be cleaved, and it is cleaved by some of the nucleases present in the nucleus. So when the nucleus work, it generates a 5' OH group. It is an unusual 5' OH group because in the 3' there is formation of OH group, and in the 5' there is formation of phosphate, but here there is formation of 5' OH group, and in the 3' end there is formation of 2' and 3' phosphate group which are in the form of cyclic structure. Okay, so next the enzymes which are known as phosphodiesterase and kinase work. What was the work of kinase? The work of kinase is that it will add the phosphate. Where it will add the phosphate? In the 5' end. In the 5' end this kinase adds a phosphate group. Okay, so what is the work of the phosphodiesterase? It breaks the phosphodiester bond which was present between the 2' and 3' phosphate group. Okay, so when this phosphodiester is work, it breaks this bond. So here there is formation of 2' OH group and 3' OH group. Okay, now next, what needs to be done? This segment which is present, it needs to be ligated for the formation of a mature tRNA. So for that it needs to be ligated, which is done by RNA ligase. And during this ligation steps, this 2' OH group which is present, it will be removed, and there will be sealing of this 3' OH group and 5' phosphate group. In this way there is formation of a mature tRNA which can work in the formation of protein synthesis. This was all about how the splicing as well as how the mature tRNA is formed. Thank you for watching this video till the end, and if you like this video, please do like, share and subscribe. Thank you.